Hi, this is Eric Ruderman from Northwestern University in Chicago, coming to you live from the Eular 2024 meeting in Vienna, Austria for Room Now. I've spent some time at this meeting looking at a number of abstracts and other information that we're seeing relative to spondyloarthritis, and several questions have come up to me this week. One in particular, which I have struggled with, is whether we truly need more interleukin-17 inhibitors and more JAK inhibitors for spondyloarthritis, either psoriatic arthritis or axial spondyloarthritis. The number of trials that we're seeing, the interest from industry would suggest that after the success of the existing IL-17 inhibitors, there's a lot of interest in identifying new drugs going after this target. I'm not sure that the market is going to bear all these different drugs, but the number of papers and abstracts and the amount of information we're seeing at this meeting is pretty impressive. These include newer antibodies directed against IL-17, and in particular, one that we've seen before, Bimekizumab, which is a monoclonal antibody that targets both IL-17A and IL-17F, two versions of IL-17, which is distinct from the existing approved monoclonal antibodies that target only IL-17A. We've seen data, and at this meeting, there's data on IL-17 nanobodies. These are smaller, parenterally injected molecules that don't have some of the limitations of full-on antibodies. In theory, they're being used in cancer, and in theory, they may have better tissue penetration. Not clear that that's really the case in our diseases, and whether this translates into improved clinical benefit has yet to be seen. There are a few small molecule inhibitors that can be given orally for IL-17 and IL-23 that are early in development, nothing here at this meeting particularly, but we're also seeing some additional data on oral JAK inhibitors at this meeting, including a new JAK1 inhibitor that's being studied in ACT-SPA, and we're seeing data on TYK2 inhibitors, TYK2 being the fourth member of the JAK family. The rationale for TYK2 inhibition is that interleukin-23 in particular signals through TYK2, and interleukin-23 being an important cytokine in spondyloarthritis. It suggests that this might be a better pathway for JAK inhibition, or at least JAK family inhibition for these diseases, potentially, and I say potentially, with a reduced risk of adverse events. Some of the things we've seen at this meeting, abstract OP0195, which is a phase 2 study of Sonelokimab, a new interleukin-17 nanobody that targets both IL-17A and IL-17F. And there's also an abstract for Izokibep, a nanobody that targets IL-17A. That's being presented as a late-breaking abstract, number 0005. Also at this meeting, there's some data on Vunakizumab, another IL-17A inhibitor. That's poster 0803. And one that I thought was interesting is an abstract on TAK-279. That's a selective TYK2 inhibitor. This is a phase 2 study being presented as an oral presentation, abstract OP0138. One of my favorite things about this particular molecule, it has acquired a generic name, which is Zasocitinib, which I think is one of the best generic names for a small molecule that we've seen in a while. All of these are showing great data in psoriatic arthritis or axial spondyloarthritis. And so there's no reason to suspect that they're not effective molecules. I think time will tell whether the TYK2 inhibitors are not only effective, but potentially safer than the existing JAK inhibitors. But I just don't know if we're going to need this many agents targeting the same pathway for these diseases. We'll see. And I think ultimately, once these trials are run through into phase 3 and hit the market's going to tell us what is necessary. So that's my thoughts on this, coming to you from Eular 2024 for Room Now. Stay tuned for more information from this meeting.